My friends, my family, this is the moment we've all been waiting for. My name is Joyce. I am your talk show host here at the Carbon Flip, where we are converting our conventional farm into an eco-friendly microbial farm. Well, what that means is we're no longer going to be using the synthetic fertilizers to grow our food. We're going to use the eco-friendly amendments that I create myself in real time right here on the farm. So if you'd like to know more about that, stay tuned. You'll get to follow along real time with me and all of my mistakes I've made so far. We do have a lot of progress as well that we'll be showing you. And for this video, we're going to look into the microscope and we're going to determine whether these microbes that we're adding to our vermiculture bins are killing the biology within the system. Now, this is a predator-prey interaction, supposedly. I'm kind of just going off of people's word right now because I'm trying that this beast is looking me in the, is behind me, and I have got to get ready for crops going in and get these amendments made so that we can begin the 2024 growing season. Here now I'm showing you a picture of the microbes under the microscope. We're at 400 total magnification. And what this is assessing is what life is still in here after I have added the mosquito bites. It is supposedly a bacteria that will kill the fungus gnats. Now, I'm okay with that. This is a system that we are I call it a menagerie because it's a confined system that we are manipulating ourselves as humans, and we want a product that's not going to harbor those fungal gnats. It's the same as with the soil food web. We do not want the root knot nematode, or no, we do not want the root knot nematodes in our soil. And so we find those predators that take care of them so they're not destroying our plants. So in this product, we want to sell, or not sell, I'm sorry, can't even sell that. We can't even say that. It was a mistake by me. As far as I know, we can't. Anyway, it's just beyond me. I don't even know what I'm talking about right now. If we want a product that doesn't promote those fungal gnats, we need to eliminate what it is that they're feeding off of. So when we create this verma bin as humans, okay, that's not a natural environment anymore. And things that we do to feed those vermi, can create a situation where, say, oxygen is depleted and bacteria goes to take off because we've added too much, say, food source. There's a number of things. We add too much water, we flood the system, it goes out of whack, pH is off, you name it. So we're trying as hard as we can within this confined system to maintain environmental, the status best of our ability. So we want our moisture, we want our oxygen, we want our food sources, we want our pH, everything to remain as close to a natural environment as possible, but that can get thrown off at any time. So if we feed these feed them too much, then that's possibly when the suppose fungus starts to grow like wild. And then the gnats will feed on that and lay their eggs because it's a perfect environment for them. So we don't want to create that environment. But in nature, there is always a checkpoint, it seems. So when I raise my nematodes to eliminate the grubs or the wax moths, because I don't want to harbor wax moths in my beehives, so I grow the nematodes, I put them in the soil so that, or small hive beetles, let's use those as the example. They lay their egg in the hive, They go wild when they get a hold of that pollen, and all the larvae come out the front door, go into the soil, and then they multiply. So I have made, this is wild. I have made the nematodes to add to the soil so that when the larvae go in there, they infect the small hive beetle larvae and it kills them. Okay, that's the same example we can use here with this, I think it's called BTI maybe, bacteria. We're adding it to this bin. We're only going to kill the specific fungus. And then there are some others that it does infect. I mean, it's not the fungus it's killing. I'm sorry. It's the fungus gnats. So it goes into the digestive tract and kills them. Okay, it's a lot of technical information that I can't quite bring to you right now because I don't have the time to sit down and pull up all the studies. And maybe I got a crew out there with time on their hands wants to do a little scientific research. And you can maybe link in a video here so that you can explain all that in-depth science behind it. I'm just giving you a very broad overview of why I feel like it's okay to add that to these bins. And we're here to show you right now with our goggles on our microscope what's going on after it's been sprayed into these. This is my vermicastings. I have removed it from my breeder bins. It's off to the side. It's maturing, which means these microbes are beginning to grow more. You will want even a 6 months mature to get that biologically complete status where it's got the most diversity of microbes possible in here. We also add different substrates that different microbes feed on to help increase that diversity to reach that biologically complete status. Okay, so yes, manipulation happens in order to achieve that. We're not just going to probably get that growing on cardboard, one single substrate. So we need to add several substrates to the vermibin to get that diversity up so that we can reach or achieve that status. Okay, beautiful. I hope I've gotten all that information out there. This is a test state amoeba. She looks very live, so I'm going to assume it has not killed her. I don't know if they think they have male, female, or they could be both. Who knows? Who cares? I'm trying to get this done for you guys as quick as possible. I'm just going to slowly, kind of look through to see what I can find. Our biology seems to be moving, growing, seems to be active in here. And I haven't done anything major. Castings may be a little bit dry. Okay, so we got to factor that in and think that possibly, see the cysts, possibly a lot of our biology is starting to go dormant, which is what I want. If we slowly bring them to a dormancy stage, I believe we can dry them down, and this is going to be an amazing way to apply vermicasting using, say, maybe just a regular old fertilizer buggy. Spread it on at a certain rate per square foot. It's going to work beautifully. Promise. Don't down me. I know it's going to work. I'm feeling very confident that I don't have a bunch of dead things here in my vermicle compost. I'm not going to take a huge amount of time at this moment, but any of you who have looked under the microscope a lot, that's what it takes. Oh my gosh, the number of samples that you have to look through in order to see differences is mind-boggling. So we have a piece of fungal strand there, but we'll be looking for more spores. And this is very young castings. This is not really matured very long. So we would have to come back months from now and reassess that. Mm. Definitely life in here, but I admit, you guys, I think it needs to be more wet. But I'm new at this, I'm not an expert soil assessor just yet. Give me time. Check back next year. I got to get through the program complete. I'm just a farmer here that thought I was getting a scam. So, as a scientist, I had to know. What what are you talking about, Willis? Um, and as I got to digging into it deeper and deeper and deeper, and as you can all see, I'm up to my eyeballs with the worms. And I think it's making some sense. It's clicking, it's clicking. Um, I believe the underground network that these plants create with this biology, I really just feel like I need to write the word stupid right across my forehead because it never dawned on me that this was happening underground. I'm a beekeeper and I know full well what happens above ground with the flower and how the plant gives off a product, nectar, sugar, to get the bee to do a job for it, pollination. Why wouldn't it be feeding the microbes in the soil sugar to get a job for it, to do a job for it? All, bringing it food, bringing it nutrients, bringing it minerals, bringing it vitamins. I'm like, oh, boom. They figured it all out. So to me, the falling away happened back when the synthetic fertilizers were created. That was the great falling away. And now we're oh, with so much more knowledge. It is so mind boggling to me, and the life and the energy that it has brought with it for me, and possibly my plants, and then the animals that feed off of it. Holy moly, you guys. Get your ship sailing and get ready. Find whatever soil you can get or dirt you can get your hands on and convert that into soil. I'm not seeing any huge changes with spraying this bacteria in here. What I need to do, though, is more of a 6-month type experiment on what happens after, or yeah, actually, I need to probably spray some in my breeder bin. And I need the mature castings, which I don't have now. I guess I could grab those out of my grower bin and assess it, and then spray the bacteria and assess it again. But I mean, knowing what I know about the nematodes, and I'm sorry, but when I tried to get them to infect other things, they didn't do it. So I'm pretty sure this is the same way, like some bacterias are bad, some aren't. And it's species specific as well. I do know a lot about that. Oh, look there. I believe that's a fungal spore right there. Darn, it's really hard to focus in on her. And this slide is a little bit busy. I need to dilute it down some more so I can get some better, you know, view on it. Okay, you guys done kind of looking through this here. This is not a tea at all. I could run an extract on it, liven them all up again. As far as your flagellates, and they like more of a moist conditions, and these I'm drying down because I'm wanting them to be able to spread onto the field this spring. I want to get this biology out there. So I'm going to do some liquid amendments too, even though I don't know what the heck I'm doing. Surely I can't destroy my plot. I have to turn one of my plots has to turn from dirt into soil in order to graduate. So this is how I plan to do it. Gonna apply the castings and liquid amendments for my thermophilic piles. Here's another spore of some sort. So that's not active though. Okay, zippity doodah. That's it for today, everybody. I have got to get out into the bees. I'll try to get a video up of what they are looking at. It's an amazing year this year. Warmed up so nice, so early, and they started laying, and they are huge in those colonies. We'll be splitting soon. I'll start grafting some queens. And hopefully 2024 is a amazing year. I'm seeing a lot of biology here. A lot of diversity. Nematode. Good thing I kept looking, right? Because y'all wanted to know if she would be dead. I'll tell you, you can just barely touch this microscope and it moves on, especially at the levels that this is. Oh, she's alive. Let's see if we can focus. She's holding real still. Now, when this light stays on her very long, she's going to really start getting upset. So I'm going to say we are bacterial feeding nematode. Which she's probably just high on the hog right now, eating all that bacteria I'm applying for those fungus gnats. Oh, she's a fat one too. I'm just not going to get a very good view of her. Here we go, right here in her mouth. I see a long cylindrical. The lips aren't huge. I'm not showing bacterial feeding omnivore at all. Oh man, can I get a good, super hard to get a good view on. This is a strictly, it looks to me, bacterial feeder. I'm not seeing a huge opening on the mouth, but more cylindrical. She's fat. Nice. She's going to give us a nice view right there, right now. Nice cylindrical mouth. Yep. No terminal bulbs. I'm not seeing any spears. And she's just not real clear for some reason. It's got to be the lighting. But it's too cloudy because I didn't dilute it down enough. But I'm not seeing a real resemblance of anything other than bacterial feeding. That's good enough. We've got life there. Okay, well, that took up more time, but isn't it just super fun to look and see what all we can see? This would really come active. There's testate amoeba with some stuff hanging out. So she's definitely active right here in the center of our screen. Sure. Right here, she got that plasma coming out. So can't see her. She's not dead. Awesome. All right, everybody. Have a wonderful day. Stay safe. I hope to see you all in the future.